Professor Noakes, you were criticized quite strongly on the basis that your recommendation of putting a child onto a low carbohydrate, high fat diet as a weaning diet is a ketogenic diet, and that may be potentially dangerous or harmful for a child. Can you comment on ketogenic diets, the ketone physiology and biochemistry and metabolism?
The first slide is just to remind us how do we develop ketosis. So what I've got here is a diagram with the liver on the left, which has lots of glucose in it, and that's called high glycogen. So this is a post-absorptive a person who's eaten lots of carbohydrates and stored the carbohydrates in the liver. And as a consequence, glucose then becomes the main fuel for both the muscles on the extreme right and for the brain. The muscles also release lactate, which can be used by the brain. And because they are high insulin and low glucagon, those are the two hormones secreted by the pancreas, there's an inhibition of triglyceride breakdown in the fat cells. So the fat cells are inhibited, they're storing fat, they're not releasing it. If you take that person off the high carbohydrate diet and starve them or put them on a low carbohydrate diet for any period of time, then the metabolic profile changes. And now what happens is that because you've released the insulin brake on the fat cells, triglycerides in the fat cells, which is glycerol and fatty acids, the fatty acids are released, they go to the liver, and the liver starts to produce ketone bodies. And those ketone bodies can then be used by the muscles or the brain as a fuel. Under those conditions, lactate and glucose become less important metabolites both for muscle and for the brain. So that's the effects. Now, this is not an uncontrolled system. It's homeostatically regulated. And I think that's a point we need to make right from the start. The human body is a homeostatically regulated system. And this doesn't go out of control. As long as you have some insulin, this system will be regulated, and the level to which the ketone bodies rise will be a complex relationship, and it'll be very individual, but it will be regulated. It's not as if these ketone bodies are just going to go rising, rising, rising. If you have diabetes, type 1 diabetes, where you're not secreting any insulin, then there is no regulation. And then what happens is there's no regulation on triglyceride release from the fat cells. The free fatty acids go uncontrollably elevated, and the ketone body levels start to rise, and they just go up. There is no brake, nothing to stop the ketone bodies going up to 30, 40, 50. And once you get ketone bodies in that range, because they're acidic, they drive the body's pH down. And the pH you need to regulate within a tight range for your heart and for your brain and your muscles to function. So in the absence of insulin, the complete absence of insulin, this is a very dangerous situation because ketosis, severe ketoacidosis develops, and that is a separate condition. I showed you my own data, and I'm going to come back with more data, that in my state at present, when I have enough, I've just got a little bit of insulin, my ketone bodies very seldom go above 1, which is nutritional ketosis. Nutritional ketosis, 0 to 2 ketone body levels. If you had a ketogenic diet where you restricted the protein and you had a very high fat diet, you were treating a person with cancer or with brain cell cancer or with repeated epilepsy, you would try to get the range between 2 and 5, and you would struggle to get it up to 5. That patient would have to be incredibly compliant to the diet, and they would never get above 5, and they would not have a ketoacidosis. In other words, if we measured their blood pH, it would be completely regulated. That is because they have insulin, and as long as you're eating, you will always be secreting insulin, because one of the stimulus for insulin secretion is just eating. So now, and so that's the range. So nutritional ketosis is what we talk about, and that's the diet we prescribe in both the Real Meal Revolution and to a much lesser extent in raising superheroes. It's nutritional ketosis. We do not prescribe the strict ketogenic low carbohydrate diet. That is a medical diet. We do not prescribe it. And then you have ketoacidosis, which is a fatal condition in patients with type 2 diabetes. It's terribly important to understand there are three categories. And when you talk about a ketogenic diet, in a sense, starvation and a low carbohydrate diet is ketogenic, but it's in a controlled system. So I said that Mrs. Leemstra should wean if it was her child. The advice was for everyone to wean onto a low carbohydrate, high fat diet. Ketogenic did not appear in what I said for a very simple reason, that the diet we prescribe, although it produces ketones, is not the ketogenic diet that you use to treat patients. And it's most definitely not the ketogenic disease ketoacidosis, which can only occur in type 1 diabetes. So I hope I've made the point that there are these three different groups. And in the books I write, we're dealing with the people who are eating a low carbohydrate diet and who have nutritional ketosis. And I'm going to show you that nutritional ketosis is present in babies right up to the age of 8 months. They have ketone bodies in the range of nutritional ketosis that we will find in adults eating a low carbohydrate diet. So to reiterate, diets that cause ketosis, there's starvation or low carbohydrate, low protein, high fat diets. Very important that they should be low protein, and that's why the diet that's prescribed for the treatment of cancer and for the control of epilepsy is a low protein diet. I'll show you why, because as soon as you eat protein, you secrete insulin and you inhibit your ketosis. So the only way to get really high levels, ketone levels, in a healthy person who has insulin has to be low protein. So protein causes the insulin secretion that abolishes ketosis. Hence, only a very low carbohydrate, low protein, high fat diet produces significant nutritional ketosis up to about 2 millimolar. And again, I want to emphasize that I don't speak out of reading the textbooks. I actually did the research ourselves, and I started in this research in 1976. And so I don't talk about it as if it's an airy-fairy concept. I actually understand it because I've been involved in researching it.
Now, Professor, on just in that area, would you say that the low carbohydrate, high fat diet, is it a very low carbohydrate, high fat diet?
The very low carbohydrate diet I describe as below 25 grams. And even at 25 grams, if you're eating protein, you will not have significant ketosis. By that I mean you won't have ketone blood values above 1. So because if you're eating protein, you can eat a very, very low carbohydrate diet, but as long as you're eating protein, you're going to stimulate insulin and you'll inhibit the ketosis.
Yes. When you tweeted to Ms. Leenstra, did you say very low carbohydrate, high fat diet?
No, it said LCHF. And I used the word LCHF because she understood LCHF because that was the question she asked. So therefore she had to have a reasonable understanding of what an LCHF diet was.
And did you suggest to her that she should have a low, very low carbohydrate, high fat, high protein diet?
I did not.
Professor, therefore, if from what you've said in respect of what can induce the kind of ketogenic diet for therapeutic reasons, one could never imagine, or could anyone assume, that you were suggesting a therapeutic ketogenic diet for Ms. Leenstra.
Only people who didn't understand ketosis would have made that assumption.
Can we move ahead then to page 627?
So these are two papers that we published early on in my scientific career. The one on the left was published, I see it's received 1979, and the paper on the right was 1980. And I've been through the evidence on the post-exercise ketosis study, and I used that to show you my own data showed that I was insulin resistant already at the age of 28, despite the fact that I was running marathons and was extremely lean. And then the next study was to see what happens when you eat protein. Alanine is an amino acid, which is part of the protein, glucose and starch, on the ketosis produced by exercise and by starvation. So already in the 1980s, we were questioning what causes starvation ketosis or exercise ketosis and what reverses it. And the next slide comes from the second paper. I showed you the slides on the first paper, and I remind you that when I showed you the first slides, my ketone bodies went up to a value of about 3 after 24 hours fasting, in which I'd run for 2 hours. I'd run 25 kilometers. I'd fasted for 24 hours, and I hadn't eaten anything, and I'd been eating a high-fat diet for 2 days before that. That's what induced in me a ketone levels of 3. And those 3, remember those 3 are below the air values that we'd sort of use for the ketogenic diet that we might use to treat cancer. And it's way below the values you see in diabetic ketoacidosis. So this comes right from that paper, and it's one of the figures. And what happens here is we use the same model that we pioneered with ourselves. We had these young men eat a high-fat diet, no carbohydrate diet for 2 days, and they came into the laboratory and they exercised for 2 hours, and we measured their ketone bodies, and this is 3 of the groups, and there's a 4th group here which we'll discuss. And what happens during the rest of the day, their blood ketone levels rise. And notice they start at about 0.5, which is my value on most days. That's a typical value for a person eating a low-carbohydrate diet with some protein and not excessive fat intake. And they rise up to, because I'm not showing you, but they rise at about 10 o'clock or 11 o'clock, they reach values of 2. That's the red curve going up on the top figure. And if they don't eat anything, you'll see that nothing changes. Their ketones now have reached a plateau and there's homeostatic regulation. And they will stay at that value of about 2 probably until the people eat again. We then fed them glucose or alanine, which alanine is a protein. And what you notice with instantaneously the ketone bodies fall. And so that within 2 hours, at about 1 o'clock in the afternoon, they're back to this resting value. So that's how quickly you obliterate ketosis if you eat protein, or in this case, glucose. And that's why it is incredibly sensitive to what you eat. So remember, these people have not eaten carbohydrates for 2 days. And it just takes a tiny amount of glucose and they obliterate the ketosis. So to develop ketosis is difficult. It takes effort. And the bottom showed that starch had a very poor effect on reversing the ketosis. But that's not the point of the slide. The point of the slide is the minute you eat, and if it has some glucose and some protein in it, you're going to obliterate the ketosis. And I will argue that this ketosis is central to human survival. If we didn't have this response, particularly in our infants, the human race wouldn't exist, because we've lived in environments of food insecurity for millions of years, and the only way we survive is within a short time you can turn on your ketone metabolism and spare the red body using glucose, and so you use ketones as the main fuel of the brain function. And this is a technical slide, but it just shows that we measure things. Here's immune reactive, this is insulin. And just to show you again, we were thinking, and this is glucagon, and these are the 2 hormones regulating ketone body production. I'm not going to go through it. All I'm trying to show you is that in 1979, 1980, we understood ketone body metabolism. We were at the advanced area of looking at this nutritional ketosis. We'd found a way of accelerating nutritional ketosis by having people exercise. We were asking questions, what hormones are involved, and we published papers in the major scientific journal in the world. And I've held that interest in ketones. And last night I woke up in the middle of the night and realized, of course, there was other work I was doing at exactly the same time. Because at the time I was helping Professor Kuslag with his research, I was doing my own research and doing my own PhD in medicine. And the question I asked myself was, what substrates does the heart need to work best? And the model I was using was a little rat heart model where we would take the heart out of the rat and we would use it in a system where the rat heart, we would study its function as if it was still in the heart, in the animal. And so we would keep the hearts alive and we would make them work as hard as they possibly could. And we would give them different fuels. And I started off with glucose, was the first fuel I used. And then the next thing I added was ketone bodies. It was the first experiment I did, was with ketone bodies. And guess what? As soon as I gave the heart glucose and ketone bodies, it worked better. Because ketones are a crucial fuel for muscle function. So I suddenly realized last night, well, obviously you always thought ketones were good because the first experiments you ever did on your rat heart, they worked. The ketones worked so well. And one of the interesting points about why that's such a great substrate to use in an experimental model is they're water soluble and you can just add them into your solution. They don't need proteins around them to travel, that they're not lipid soluble, water soluble. And that's why they're so important in humans, because they're water soluble and therefore they can transfer around the body with great ease. They don't have to cross membranes and be built with proteins protecting them to carry them. That's why they're such an important fuel, because they're water soluble, but they act like a fat. And so my point was that not only were we doing this research on ketones, but I was using it in my own work for my own PhD on heart metabolism. And I was studying, and I was measuring how much ketone bodies are being used by the heart. So I was measuring ketone use by the heart. So again, just to show I'm deeply embedded in ketone research. And that's not discovered, so I can't show you, but there's the study of ketone bodies in the heart. And then one of my PhDs was one of the very best PhDs any of my students, Sandy Welton, did this study, and we again put people on high-fat, low-carbohydrate diets, and we studied their metabolism, and we infused insulin, and we did all sorts of other exciting studies. Some of the best studies showing that the content of glycogen in the muscles, not just the liver, influences the body's response to exercise. And when the body is carbohydrate depleted, it's converted into this fat-burning state. And we were able to show in this model that the key was the muscle, how much glycogen or carbohydrate was stored in the muscles. And I'm not going to go through the detail, but the point was that we did good research. We're not going to go through it. If anyone is interested, just to show you that these were the conclusion of those studies. They are very, very complex studies. And they were at the cutting edge of understanding why does a body turn from carbohydrates to fats when you restrict carbohydrate ingestion, carbohydrate intake. So here now we've got some basis for looking at where does energy come from for the brain. So the liver produces the glucose, there we go. So you eat carbohydrates, the carbohydrates comes into the gut and it's absorbed and it's stored in the liver. And that provides the obligatory glucose need if you're on a high carbohydrate diet, and it's 125 grams of carbohydrate per day. And that is why people say you need 125 grams of carbohydrate per day, because if you're eating a high carbohydrate diet, that is your obligatory glucose need. The brain needs to burn a certain amount of glucose. There is an obligatory need. For most people, it's provided by what they eat in the diet. But many of us are eating much less than that, 125 grams, so where's it coming from? So if you're not eating dietary carbohydrate, where's the glucose coming in the bloodstream? And the answer is, firstly, ketone bodies are released, particularly when you're starving, as I've shown you, or if you eat medium-chain triglycerides, you increase your ketone bodies. And they then become an important fuel for the brain. And they are critical for brain function when there's a low-carbohydrate diet. Lactate is used when you do high-intensity exercise, or interestingly, after a brain injury. Lactate suddenly becomes a crucial fuel for brain injury. And I suspect ketones are also in brain injury. We'll discuss that, but that's not yet proven. Proteins come in and fats come in, and that produces the glucose by the process called as gluconeogenesis. And so that's where you get your glucose if you're not taking any carbohydrate in. You get it from protein fat in the liver, converted into glucose in the process of gluconeogenesis. Now, when you're fat adapted, as I will show you the study of Owen in 1967, your obligatory carbohydrate intake drops to 25 grams per day. And the liver then is perfectly able to generate 25 grams of carbohydrate per day. So any dietary guidelines that say you need 125 grams of carbohydrate per day is true if you're eating a high carbohydrate diet. If you're eating a no or low carbohydrate diet, you need 25 grams, and that, as I will show you, is provided by protein and fat in the diet. So there is plenty of glucose. So the point of this slide is that you do not have any obligatory carbohydrate need to eat carbohydrate. We have a liver, and the liver will generate all the glucose you require to keep your brain functioning. And again, to make the point that the primary defect in diabetes is gluconeogenesis is increased. That is the primary driving abnormality. And if you have increased gluconeogenesis in type 2 diabetes, why would you need to intake in glucose? You wouldn't, because you're already overproducing glucose.